carbon. I'll first go through the forms of carbon, then I'll talk about how the forms transform from one to another, and then I'll give you at the end a general introduction to nutrient cycling, how we're going to diagram nutrient cycling and diagram the carbon cycle. So the first of the things that you need to memorize up front. So we have carbon dioxide, and almost immediately when carbon dioxide enters water and dissolves, it forms carbonic acid. Okay, so just the action of CO2 dissolving in water forms an acid, and you get this decrease in pH. So we already talked about coral reefs being endangered because of increases in atmospheric CO2. It's because the oceans are acidifying because of that. It's forcing it into dissolve in there. And then once it gets dissolved, we have carbonic acid, bicarbonate, and carbonate. And this leads us to the bicarbonate equilibrium. And I just leave the, we don't have to worry quite so much about carbonic acid or CO2. We can think of those kind of as the same thing because that's a very quick transition. But then we have the equilibrium with, you lose a proton, you get to bicarbonate, and if you lose another proton, you get to carbonate. This is an equilibrium that you've probably experienced before. It's a function of pH, and we see too that the carbonic acid or CO2 dominates at low pH, that carbonate dominates at high pH. Practical example of this, if I took a can of soda or beer and I injected somehow a strong base into that, what would happen when I opened the can?
Well, it may explode.
Nope, exactly the opposite. If I put a strong base in. So are beer and soda, are they acidic or are they basic? They're acidic, right? So they're acidic, they're forcing the equilibrium to this side, and the CO2 is supersaturated and it's held in there under pressure. So when you open the top, the carbon dioxide comes out of solution and bubbles up into the atmosphere because it's supersaturated compared to what it should be. If you took that can and injected the base in there, that strong base, it pushed the pH over here, the predominant form would no longer be CO2, and so your beer would be flat. It would be the saddest thing that could happen, perhaps. Some of you also have done the experiment in science fairs or whatever, where you make the volcano and you take baking soda and you throw vinegar on it. Basically, you take this stuff, you throw the acid on it, it forces equilibrium to this side, and it puts CO2 out. So looking at that as a chemical equilibrium, essentially, if we add protons here, it forces the equilibrium to this side, right? If you remember your chemical equilibrium, it's all got a balance. If you take protons out, if you add hydroxyl ion, it uses up those protons and turns it into water. It pulls the equilibrium over to this side. Okay, so that's why it's called the bicarbonate equilibrium. That's also why bicarbonate, if it's in substantial concentrations, buffers water against changes in pH, because it's basically reacting with these protons and putting them into the molecules as opposed to being out in the free water. That's part of the basis between the alkalinity assay. We're doing that on Tuesday? Tomorrow. Okay, we'll do the alkalinity assay tomorrow. And the way you do it is you essentially titrate with a known acid, and you see how much acid it takes to cause a set change in pH. This will also become important when we talk about acid precipitation, because systems that are buffered against pH changes, those that have a lot of bicarbonate in solution, are much less sensitive than those that have very low bicarbonate concentrations. So I promised we'd talk about death and destruction. In Cameroon, 1,700 people died in 1986. And one of the survivors reported a 25-meter high surge of water going up the side of this thing. And this caused by a catastrophic release of supersaturated carbon dioxide from the hypolimnion. They think that the carbon dioxide came from volcanic activity underneath the lake. And because the hypolimnions down, won't mix, right? That's part of what a hypolimnion is. And there's a high pressure down there. You can get a lot of CO2 dissolved in the water, right? Very high concentration of CO2 dissolved in the water. And then there was a landslide or enough weather to cool the epilimnion down to match the hypolimnion, or a combination of both those cool weather and the landslide that allowed that to start. And once that supersaturated water came up to the surface, it started degassing and letting bubbles come out, and that caused the whole lake to turn over. And it's like when you open a pop, like how it foams over, it surged up the side and let that CO2 go. So now the water's building up again, and they're using pipes to release the pressurized water. And the French came up with this. It's a really cool way to do it. They take a pipe, they put it down into the bottom, they pump water up into the surface, and when it gets up here, it's all of a sudden supersaturated. It's releasing pressure, so it starts releasing its gas. And it starts releasing its gas into solution, and this actually makes for an airlift pump that keeps pumping the water up without any energy input after that. Right, so it's a really ingenious solution, actually. And this is a picture of the first one that they put in there. That they have about a 50-foot high jet of water coming out of this thing, and that's not from any pumping they did. They started it by pumping it, but then they just let it go, and it just, because this gas coming out of solution so violently, it shoots the water up out of the bottom and continues to mix the lake. So I think they've got this solved, because they've got a number of these throughout the lake that are continuously releasing the CO2 pressure. So if it turns over again, there won't be nearly as much. This is an example of physical limnology in action. Understanding the chemistry of it led them to a fairly nice solution that hopefully will keep this from happening again. There are other volcanic lakes that have similar things, and they've seen similar problems, not on that scale, also nearby in Africa, but also in Iceland has had a number of livestock deaths associated with this kind of event. Okay. Now we'll move into something that's a little bit more complex, organic carbon. And again, as with all chemicals, we can make this distinction between dissolved and particulate. But if you remember that figure I put up with the spatial distribution of compounds, it's a artificial distinction, because it's based on what filter we decide to use to filter it out. Usually it's 0.45 microns is the line between solid and particulate, but it's just because that's what the filter we have. You can also think of it as coarse particulate organic material and fine particulate organic material. This is what the people that do stream research, this is how they classify things. So coarse particulate organic material might be leaves, branches, fish. These don't get counted. Maybe if they're dead, they count them. They technically count, but you usually don't count them. Fine would be all the fine sediments in the organic material associated with the fine sediments. Again, usually dependent upon some sort of arbitrary mesh size that that's fairly agreed upon, but there's no real reason for that particular size. Another type of way to talk about carbon is biochemical oxygen demand. And organic carbon is a big part of biochemical oxygen demand, although it's not all of it. There's some other inorganic chemicals that can have oxygen demand. And essentially the way this assay works, it's an old assay that's used to assess the effectiveness of sewage treatment. You take some water, and if it's sewage water, you'll dilute it out. Put it into a BOD bottle, which is we those. And then measure how the oxygen changes over a period of a couple of weeks. How much oxygen demand is there in that water? And so basically it gives you an idea of, in this case, if you dump the sewage into a lake or a river, are you gonna cause the lake or river to go anoxic because there's too much biochemical oxygen? So it's another way of thinking of it as bioavailable carbon. And then there's all kinds of complex things. Essentially any organic compound that animals or plants synthesize when they die goes out into the environment. Some of them break down extraordinarily quickly, right? So proteins, DNA, because the organisms have a big demand for them. Others, particularly the structural materials in plants, take a long time to break down. General method, again, for diagramming nutrient cycles, and then we'll lay in the carbon cycle over that. So, oxidized inorganic form, reduced inorganic form, and organic C, in this case. And so we essentially have what we can consider a typical, and this is similar to a typical ecosystem diagram, in that we have boxes and we have fluxes between the boxes. And if we have oxygen from the midline up, and we have anoxic from the midline down, if we take a reduced to an oxidized compound, when oxygen is present, is that going with or against potential energy? With, right? So we go with potential energy. And if we take an oxidized to reduced, in the absence of oxygen, is that going with or against potential energy? Which? That's going with potential energy, right? So that's the idea of the solution you're in, determining what the potential energy is of the chemical you have. And then we always have these organic molecules that are sort of recycling themselves. That's how most heterotrophs do most stuff, particularly more complex animals. They don't take up inorganic nutrients straight up for the most part. They get them from their food in the form of amino acids or nucleic acids or phospholipids or those kinds of things where the phosphorus is already part of the lipid. We can't just take up phosphate and incorporate it. Right? Whereas a lot of the autotrophs and the simpler, the microbial, particularly the bacterial, can use things like ammonium and nitrate and carbon dioxide and those kinds of things, the inorganic forms, and turn them into the organic forms from the bases, and the plants as well. They can do that. Okay, so that's a generalized diagram. And we can take that diagram and we can use that methodology to start by plotting the organic carbon cycle. So we'll still have the organic carbon here. And we will have carbon dioxide being the oxidized inorganic form. And then I'll put methane in as the reduced inorganic form. It is an organic molecule, sort of, but it's just easier to do it that way and more explicit. And again, we have oxygen here, lack of oxygen here. So if we take carbon dioxide and we transform it to organic carbon in an oxidized environment, is that going with or against potential energy? Against, right? So what kind of organism is doing this? Photosynthetic, right? And what other type of organism can do it? Chemoautotroph, right? So it's the autotrophs. carbon dioxide. When we get to the sulfur cycle, we'll find out that there are organisms that can photosynthesize anaerobically as well. And they actually don't generate any oxygen. That's anoxygenic photosynthesis. We'll get back to it and I'll describe it in more detail, but it's a key, it's one of the fluxes. Okay, so if I take methane and convert it to carbon dioxide, am I going with or against potential energy? Right, because I'm up above, I'm in the oxygen area, right? So I'm oxidizing methane. Organisms that oxidize methane and generate energy, what's the name of those organisms? Ones that eat methane, if you want to think of it that way. Okay, what's the first part of the root for methane? Methano, right? What's the second part of the root for eating things? Troph, right? So methanotroph. Okay, methanotrophy.
So you're saying that's going with the gradient?
Right.
Okay.
So those are the organisms that are sitting right on top of the sediment at the oxygen-oxygen interface, just on the oxidized side, waiting for that methane to come out and chomping it up, right? Eating methane. Now, we also can have organisms that take carbon dioxide in anoxic environments and going towards methane. Is that going with or against potential energy? With, right? Yeah. Okay, so that's going with potential energy. This is methanogenesis. Okay, so they're generating methane, methanogenesis. Okay, so methanogenic bacteria are doing this in very low redox conditions. We also have organisms that are converting organic carbon to CO2 in the presence of oxygen. What's that process called?
Respiration.
Respiration, right? It's going with potential energy, and it's called respiration. The last bit is that organisms, in the absence of oxygen, are going to either respire organic carbon with alternative electron acceptors, such as nitrate or sulfate, or they're going to rearrange the organic compounds using the oxygens that are in them to gain energy through the process of fermentation. Right. So fermentation can either just create another organic molecule, and it will also push off some carbon dioxide. So like when I have my beer brewing in my basement and I put the yeast in, the oxygen goes away and it's bubbling away merrily. They're throwing off some alcohol, but they're also throwing off carbon dioxide. So the alcohol they're throwing off is here, organic carbon, and the carbon dioxide they're throwing off is there. We also mentioned that some methanogenic bacteria can use acetate to generate methane as well, and we can classify that as a fermentation if we want to. So you can also generate methane through fermentation. So all of these are fermentation. And finally, as I started this, I told you this idea of how we're, as heterotrophs, just basically reorganizing organic compounds all the time. That we're taking proteins, breaking them down to amino acids, and building back proteins back up, and repeatedly. These heterotrophic cycling occurs. And this happens whether you're in an aerobic or an anaerobic environment.